Bismillahirrahmanirrahim. My dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. Welcome back to AmreenaMehrajTutorials. Today I am in front of you once again with this interesting topic called genetic disorders. It's a very important topic as far as your board examinations or your competitive examinations are concerned. Questions are asked every year from this particular topic. It's very interesting. In this particular topic, my dear students, we are going to discuss Mendelian single gene disorders. So what does this mean? I'll make you understand that thoroughly today. Mendelian single gene disorders. So get this hint of the term gene disorder. So over here there will be some defect in the gene, okay? And the other category which we'll be discussing will be the chromosomal disorders, okay? So there will be two broad categories which we'll be discussing. It will not be completed in just one video lecture. It will take us a couple of video lectures to finish all the disorders. So there's a hint, gene. So there will be mutation in a gene. Chromosomal, there will be chromosomal aberrations. Now what that means is basically clear to you people from my last 5 video lectures, okay? So our topic, our aim is to discuss many Mendelian single gene disorders. Suppose under this category, we'll be discussing hemophilia, phenylketonuria. For some idea you people will get. Chromosomal disorders, we'll be discussing Klinefelter syndrome, Turner syndrome, etc. These disorders we'll be discussing in this particular category, okay? So this topic is going to be basically a vast topic. I'll be trying to give you advanced and standard information so that you will not feel any difficulty if you will consult any of the standard reference books, be it from foreign author or be it from some Indian authors, okay? So my dear students, this lecture is going to be very fruitful, very beneficial for all of the students. So don't forget to like, share and subscribe to my channel and comment also, because that's very important. From your comments, I get some kind of, positive, thrust, and I feel motivated, and that drives me to make more videos. So your comments are basically the driving force, and your comments make me understand what you basically want. So I want you people to type in the comment section below which is the genetic disorder you want me to make a video the next, okay? So I'll be making next video out of your suitable suggestions. It should not be a random suggestion. If it will be a suitable suggestion, I'll be making a video on that, okay? And while I'll be delivering this lecture today, I'll be asking certain questions. You are supposed to type the answer in the comment section below. The moment you listen the question from my side, type the answer. Then I'll be, if it will be a correct answer, I'll be giving you a positive signal. If it will be an incorrect answer, don't be afraid. I'll be guiding you. That's my job, okay? That's my responsibility. So let's start this topic, human genetic disorders. Okay, so I have some stuff which is written on the board. It's pre-written because I don't want to miss these things. These are very important, and generally I don't give so much of the importance to this stuff, but they are very important because you are asked these things, okay? So who basically introduced this term genetics? This term was introduced by Bateson in the year 1906. Who is considered the father of genetics? It's known to you people from your foundation classes, somebody who performed experiments on garden pea by Simsataibum. He studied the inheritance pattern of various characters involved in garden pea. He is Gregor Johann Mendel. He is the father of genetics. Although his work was not acknowledged, his work was not accepted at that point of time because he spoke something very different to what was basically present in that scientific era when he performed his work. Later on his work was rediscovered, okay? So but he is credited as father of genetics because he gave us the basic principles like the law of dominance, law of unit character, law of segregation, law of independent assortment, etc. Later on, it was the year 1902 when basically till this point what was done, basically chromosome was discovered. It was being understood that there are genes which are basically located on the chromosomes and they are basically transferred from one generation to another generation. So it was understood till this point that the hereditary constituency is basically what? It's basically the chromosome. So this particular person called those things, what we call gene today, he used to call them the factors, okay? Then the same work which was done by Gregor Johann Mendel, that was rediscovered and that was basically accepted as a parallel opinion for chromosomal theory of inheritance. So whatever is there in this chromosomal theory of inheritance, which was put down by Sutton and Boveri in 1902, independently these two scientists came up with this theory. It is basically parallel to what Mendel has said. So over here we have the concept of genes and alleles. He spoke about unit character, he spoke about factors and all, okay? Then later on, with further investigations which were done by an eminent scientist, we call him the father of experimental genetics. He is Thomas Hunt Morgan. He performed various experiments on Drosophila melanogaster, the fruit fly. And he then came up with this idea of linkage, what we call chromosomal theory of linkage. He gave us some idea about certain genes are basically linked with each other. They do not get separated. They do not separate or segregate. They remain, linked with each other once they are transferred from one generation to another generation. This is how genetics has been, moving ahead, how this advancement in the field of genetics was done from earlier times to the current times. It's an expanding science. There is no stop to it. There's no full stop. It's being explored every day. Okay, new concepts are being. It's an open field because we don't know everything about what is there in the chromosome. So that thing is investigated every day. Okay, so genetic disorders, my dear students, here we'll be discussing what we'll be discussing about the defective part. We'll be discussing about those mutations which are causing various disorders in humans. Okay, so under this category, this will be very beneficial for you if you get to know about this thing first. It's called human genetics. We cannot directly go to this topic because we have to create a foundation first. We have to be familiar with certain terms. Then we will be able to understand these genetic disorders. Unless and until we do not know the basic terms, then it will not be justice to this particular topic. So what is basically human genetics? In this particular field, we study what? We study the human hereditary traits. What do we study? We study human hereditary traits. Under what? We study this thing under human genetics. What do we study? Those traits which are transferred from one generation to another generation. When I use the word trait over here, you remember some genetic disorder. If we talk about hemophilia, we can say this is a trait. Okay, so we study what? We study the human hereditary traits, those traits which are transferred from one generation to another generation. We don't only study them, we study variations also. Okay, we study variations also. So we study this under human genetics, we study variations under human genetics. We also study the mechanism. What do we study? We study the mechanism of inheritance. Inheritance means what? Inheritance means the same thing, transmission from one generation to another generation. So what is the mechanism of the particular trait once it's transferred from one generation to another generation? Is it X-linked or it's autosomal linked? We study that also under human genetics. And other than that, we also study the pattern of inheritance. Okay, this is the nutshell about human genetics. This is a nutshell about human genetics. Pattern, because in certain, once you draw a family history chart, what you call pedigree, once you draw that pedigree, you will see that a trait is basically absent at certain generation, but it appears at another generation. So what is the pattern of inheritance? We investigate that also under human genetics. So we have to be familiar with a number of things before we start this genetic disorders. Okay, so human genetics, this field came into view. There's a prominent name you have to remember over here, that's called Sir Archibald Gerard. This is a prominent name, Sir Archibald Gerard. He is called the father of biochemical genetics. Okay, he is the father of biochemical genetics. Okay, let me give you an idea. It was basically this person, Sir Archibald Gerard, along with Bateson. Okay, these two, what do these two scientists do? These two scientists basically scanned a long population. They scanned a huge group of, organisms or humans, we can say, and they found out, these two scientists, they found out that there are many disorders, there are many defects which are there in humans. The reason for those defects lie in mutations. The reason for those defects lie in mutations of what? Mutations of the genes. So the reason for those disorders was what? Some kind of defect, some kind of mutation, be it point mutation, be it different types of mutations, deletion, substitution. I have made you understand about them, and I've given you idea about every kind of a possible change which can happen to your genetic repository. So these two scientists came up with this idea that there are many disorders. Say for example, Archibald was somebody who actually investigated alkaptonuria. Okay, he studied this particular disorder. So he came out with this conclusion that although this is a metabolic disorder, but the root, the cause for this particular disorder lies with the defect in the genetic makeup. So they got a linkage between the metabolic disorders and the genetic disorders, saying that the reason for these disorders, what we today call genetic disorders, because those disorders were what? They were metabolic disorders. They were related to the metabolism, defective metabolism. But they gave us an idea, these two scientists gave us an idea that the defect lies with the genes, the defect lies with the mutation which has happened to the genes. That's why these traits are appearing in certain individuals. So that's why Archibald Gerald is considered as the father of biochemical genetics. Okay, my dear students, now let me take you to this world of genetics. First of all, let me make you understand about it. So you remember this point that it was Archibald Gerald and William Bateson who basically scanned human population, and they came up with this idea that genetic disorders are caused by what? Basically the root for various metabolic disorders is the defect in the genes. Okay, the defect in the genetic material. Now, my dear students, what else did Archibald Gerald suggest? So Archibald Gerald and William Bateson, they said what? They said that the genetic disorders, what? They show Mendelian inheritance. Okay, not all, majority of them show Mendelian inheritance. Now, before going into that detail, let us try and classify genetic disorders. There are broadly two types of genetic disorders. Look, over here you will try and understand this fact. There are broadly two types of genetic disorders. Number one type of genetic disorders are called gene disorders. Pay attention, gene disorders. What is the idea you get from the term gene disorders? Gene disorders are those disorders which are caused by defect in gene. Okay, the defect in the gene, that is, there is a defect in a single gene. Okay, and the defect in that single gene follows Mendel's laws of inheritance. That is very important. So these disorders are also called what? They are also called single gene. They are called what? Single gene disorders. Because why you call them single gene disorders? Because there is mutation in a single gene. That's the point you have to understand. There is mutation in single gene, and that mutation, that mutated gene follows the same pattern of inheritance what was investigated by Mendel. Okay, so that gene is going to follow law of unit character, law of dominance, law of segregation, law of independent assortment. That entire inheritance protocol is called what? That's called Mendelian inheritance pattern. So these disorders, they follow Mendelian pattern of inheritance. So can't you call them then Mendelian, what do you call them? Can we call them like this? Mendelian single gene disorders. Why I inserted the word Mendelian? Why I use this prefix Mendelian? Because they follow what? These disorders follow Mendel's laws of inheritance. Now you could ask me a question, ma'am, that means we must be equipped with this knowledge of Mendel's laws. Now that will be a straight away deviation, but I will be making you get back to the terminology so that you will not face any difficulty over here. Okay, so we'll go to that stuff in a bit. The other category will be what? Which category? The other category will be that category in which there will not be mutation in a single gene. In those cases, you could have what? You could have basically change in the number of the chromosomes. What we call chromosomal aberration. You could have anomalies. More number of chromosome, less number of chromosome. Okay? So other category is simply chromosomal disorders. So these chromosomal disorders are going to be, the cause will be chromosomal aberrations. Very good. Have I taught you about this? Yes, chromosomal aberrations, or there could be anomalies in the number of chromosome. Okay, anomalies in the number of chromosome. They could be like +1 case, -1 case. That could be the reason for these chromosomal disorders. So over here, the reason lies with what? The reason lies with extra number of chromosome. Maybe extra, or maybe what? Maybe less X number of chromosomes. That will be the specific reason for these disorders. You will not be having any single gene mutation. In this category, you have a single gene mutation. Maybe the gene which is coding for some enzyme that undergoes mutation, and that results in the formation, in the appearance of a particular trait, a particular disorder. So that is the case with Mendelian single gene disorders. They are going to be always what? They are always going to be inherited. They are always going to be what? They are going to be inherited from one generation to another generation, because the defect is in what? The defect is in gene. So that's going to be transferred. Maybe the trait will not appear in some generation. That depends upon what type of disorder it is. Okay, so it may skip some generation. That is secondary question, but it is going to be transferred from one generation to another generation. But over here, they are not going to be definitely, inherited. Not inherited always. So it's not a general rule for these disorders. Somebody who is suffering from Turner syndrome, then that particular syndrome will follow the entire generation, entire family. No, that's not the case. Chromosomal disorders may remain confined to a particular organism in a family, but these are transferred from generation to generation. That is the basic difference between the two types of disorders. So you need to have this understanding first of all. So under chromosomal disorders, we'll be discussing two types of chromosomal disorders. So we'll be discussing those disorders in which the defect lies in the autosomes, and other disorders in which the defect lies in sex chromosomes. So first of all, get this idea how many types of disorders we have to study. We have to study those chromosomal disorders in which the defect lies with the what? Defect lies with the autosomes, okay, and those chromosomal disorders in which the defect lies with the sex chromosomes. Say for example, we'll be discussing Down syndrome in this case, in which you have one more autosome, or say for example, we'll be discussing Klinefelter syndrome, in which you have change in the number of the sex chromosomes. So this will be the category, okay. Now my dear students, before moving ahead, let me first of all make you understand two things over here that will be general for these disorders and this disorder. First of all, look at the diagram which will flash on the board right now. Can you tell me what is it basically? What is it basically which you are looking at the board right now? It's a diagram I'm trying to show you. Yes, absolutely correct. It is what? It is human karyotype. Very good. It's human karyotype. All of you got it correct. It's human karyotype. You have all the chromosomes in this image. So this image will disappear shortly. So you have all the chromosomes. Chromosome number 1, 2, 3, 4, 5, 6, 7, 8 up to 22. And then you have what? You have the last chromosome, which is XX and XY. Okay. Now all the chromosome pairs. So you have all these pairs. They are called what? They are called homologous. What are they called? Homologous pairs of chromosomes. From what? From chromosome number 1 to chromosome number 23. For time being, 23. Okay. For time being, let us take only 22. First 22. Okay. So we are taking the first 22 pairs. These first 22 pairs represent your autosomes. So there will be defect maybe in the gene of autosomes. There may be defect with the number of autosomes. So accordingly, you can have Mendelian autosomal disorders, chromosomal autosomal disorders. Getting my point or not? Number second, you are left with what? You are left with the last pair of chromosome, which is what? 23rd pair. 23rd pair of chromosome is the sex chromosome. Although we have other terms also, but I don't want to confuse you people, okay? So you have 23rd pair sex chromosome. So if there will be some gene mutation in sex chromosomes, then you have single gene, sex chromosomal disorders, or you have chromosomal sex chromosome disorders in which there will be defect in the number of the sex chromosomes. Okay. Now my dear students, the interesting thing is that a male and a female has got different sex chromosome. Rest are same, autosomes are same, but as far as 23rd pair, it is different. In male, it is XY, and female, it is XX. Okay. Now, XX is homologous again, there is no problem. XX pair is homologous, no problem at all. But XY, it is not a homologous condition. This is a It is which condition? It is a hemizygous condition. Let me make you understand what does it mean. What does this mean, hemizygous condition? I know majority of you will be knowing about this thing, but it's very important. This is the foundation. I'm trying to lay down the foundation for you people to make you understand these disorders, okay? Now, my dear students, if you look at this X and Y chromosome, let me make it over here. So this is your X chromosome, and over here, I'm trying to draw the Y chromosome. It's comparatively short like this, okay? So this is X chromosome, this is Y chromosome. Both of these chromosomes, so this is X, this is Y. Get my point over here. Both of these chromosomes, they have got a small region on which, this particular region, which I'll shade with black. This particular region is going to be that region which has the same genes at both the places. So whatever is the gene over here on X, the same gene will be there on the Y. This particular region, because it has the same genes on the two different chromosomes, we call it the homologous region. It's the condition with the male sex chromosome. It has got a homologous region between this pair, X and Y, a small region which is homologous, at which you have a definite loci on which the same gene is present. The rest of the region, the rest of the portion is the portion which is going to be absolutely different between X and Y. This portion is called the differential portion. It's called the differential region because it makes the difference between the male and the female. So these genes which are located on this particular portion, I'll label it this particular portion, which is now green. That represents which region? That represents the differential region of male X and Y chromosome. Now the third point is, my dear students, now look at the differential region of Y. Differential region of Y. This particular region. This is what? This is the what do you call it? Differential region of Y chromosome. This differential region of Y chromosome has got those genes which are exclusively present in Y chromosome, okay? Those genes which are located in this particular region, of Y chromosomes, those genes are called Holandric genes. Remember this point, this is very important. They are called which genes? Holandric genes. Why? Holandric genes are going to be exclusively present in a male, okay? Because these genes are located on male Y chromosome. And which part of the male Y chromosome these genes are present? These are present on the differential region of Y chromosome, which is totally absent in female, because the X form, which is present in female also, that contains different set of genes which are not present on the Y component, because this already makes the differential region. So in this homologous region, you will have the same genes, okay? In this case, you will have the same genes. Homologous region has the same genes. It will be better to use the term allele, but for the time being, I'm keeping it simple. So it has homologous region has the same alleles at the two places. But the genes which are located on this particular region, they are the Holandric genes, which will be present only with the males. So if there will be some mutation happening to this gene, that will be transferred. That will go only to the male offspring. That will go only to the male offspring. Can you think why? Why, if there will be some mutation in the Holandric genes, that will be transferred only to the male? Yes, you are supposed to type me the answer. Why will that mutation go only to the male? The answer is very simple. Because this particular Y chromosome will go only to the male. Male organism, we know it contributes what? What does a male contribute? Male contributes X carrying sperm, Y carrying sperm. But X carrying sperm only go to the female, because then the product will be XX female. Y carrying sperm has to become the part, it has to fertilize the egg, then the offspring will be always male. So if there is some kind of defect in the Y region, in this Holandric region, that trait will be transferred only to the males. That's called the Y linkage. What do you call this? You call this Y linkage. There are certain disorders, there are certain traits, like we have hypertrichosis, in which the male produces excessively dense hair around the air pinna. That reason, the mutation which is causing that excessive hair growth around the pinna, that condition is called hypertrichosis, but the defect, the mutation is with the Y differential region. That's why only that kind of trait will be shown by the males, because this is only going to become the part of the male individual. individual. Okay, so this is an idea I told you about autosomal. So in this category you will have autosomal, some autosomal disorders, some sex-linked disorders. In this case again you have some autosomal disorders, some X-linked disorders. Now what does this X and autosomal and sex-linked means? I have made you understand this condition where the two chromosomes are absolutely neither homologous nor they are non-homologous, because they have a small homologous region also. So they are not non-homologous. They are not fully homologous. They are not fully non-homologous. What do we call this condition? We call this condition hemizygous condition. Okay, I hope you got this point. Now, my dear students, let us try, because after getting into this insight that some disorders will be what? Some disorders will be autosomal, some disorders will be sex chromosomal. And over here I will give you one more point. Since Y linkage is going to be confined to the males only, because in this case males will be affected. So what is the sex-linked disorder we have to focus more as far as examinations of this level are concerned? We have to be focused with the linkage of X chromosome. So we'll be discussing which chromosome linked disorders. We have to discuss X chromosomal linked disorders. So we have brought two categories of genetic disorders now. Some will be autosomal disorders, some will be X-linked, or we say what? Sex-linked disorders. Okay, so you got two broad types. Okay, now let us go further deep. Why I'm not directly going to the classification? Because you need to have one more information before starting. You need to have this information. What does this dominant mean? What does recessive mean? Because we have to further categorize these disorders into subtypes. Let me erase this first of all. So human karyotype, you got this idea. Okay, so you have this two divisions now. You have chromosomal disorders and you have what? You have the single gene disorders, Mendelian single gene disorders. Okay, now again the word Mendelian, that means we will now be having what? We will now be having two categories. Let me erase this also. I'll erase this line also because I don't require this. You need to be very smart. Now we are talking about Mendelian. Okay, we are using the word Mendelian. So definitely Mendel's terms will be now used. Who has given us this term of dominant and recessive? Who has given us this term of dominant and recessive? Who has? Come on, tell me. So who has given that word, this word called dominant and recessive? Definitely Mendel, mashallah. Now my dear students, before going into the depth, I just want to remove this particular disorder from the board, because our first aim will be to remain focused upon what? Single gene disorders. As far as chromosomal disorders are concerned, over here we'll be discussing two types of chromosomal disorders. Let me give you the classification first of all. In this case, we'll be discussing about, first of all, autosomal chromosomal disorders. They're very short. We don't need to be so much bothered about these, because they're very, very easy. They're a bit complicated. So another category is sex chromosomal disorders. The simple thing is what? Simple thing is what? Basically, there is some kind of non-disjunction, a kind of defective synapses taking place, due to which the number of the chromosome is getting more or less. You have some other reasons also, some other chromosomal mutations. Go back to the chromosomal mutations, you will get an idea what I mean to say. So the chromosomal mutations are basically responsible for these disorders. In this case, you usually have what? Either you have translocation, you have duplication, or you have majorly what? You have hyperploidy or hypoploidy causing these particular disorders. Under autosomal chromosomal disorders, my dear students, we'll be discussing, pay attention, here our aim will be to understand about Down syndrome in detail. I've given you introduction to these. We'll be discussing Edwards syndrome in detail. We'll be discussing Patou syndrome in detail. Okay, so these three for the time being I'm giving you: Down syndrome, Edwards syndrome, Patou syndrome. And under sex chromosomal disorders, we have to discuss Klinefelter syndrome. They are all in your textbook, that's why I am quoting them, okay, so that you will become familiar with the classification. Klinefelter syndrome, Turner syndrome, because in these cases sex chromosome will be involved. Turner syndrome, then we'll be giving you super females. I'll be giving you idea about super males. Okay, All other cases which will arise in this thing, I'll give you that later on. So for the time being, I just want to erase this particular section because I want to remain focused with this, because in this particular category of disorders, we need to have the information about Mendel's laws of inheritance. Okay, so for these things we do not need that particular stuff, because they are not going to be in hereditary. They are not going to follow Mendel's laws of inheritance. That is the basic difference. These disorders are not going to follow Mendel's inheritance pattern. They are going to be present in some offspring because the defect was there in the faulty gametogenesis. The gamete which was formed was having basically excess chromosome or less number of chromosome. That majorly causes such types of disorders. So I'll be removing these disorders. Shall I remove them? I'll be removing chromosomal disorders. So once we'll be finished with gene disorders or single gene disorders, then our topic will be chromosomal disorders. And under chromosomal disorders, I'll be taking up and I'll be making you understand every technicality as far as Down, Atwood, Part2, Klinefelter, Turner, supermale, superfemale is concerned. Okay. For the time being, I need my board because I need to take you to this understanding further. So you got my point that single gene disorders will be now those disorders in which the mutation lies in the gene. The mutation lies rather in a single gene, okay? And that gene could be located on some autosome, or that gene could be located on specifically sex chromosome. But which sex chromosome? X chromosome, because I said Y linkage is, although that is also a vast topic, but over here you have to remain focused with X-linked disorders as far as class 12 is concerned and your competitive exams are concerned, okay? Y linkage I'll teach you separately. So let us now try and analyze. You should ask me that, ma'am, why are you giving us some further knowledge? Because we have to classify now single gene disorders. Let me tell you now how we have to classify these single gene disorders. They will be now divided into two types again. Okay, they will be further divided into two types. So you will have these single gene. Let me put down this in one box. So you have these single gene disorders. Okay, where is the other category? That is gone. That is chromosomal disorders. I have deleted that for the time being. We'll be focused with this particular stuff. Okay.
My dear students, don't be surprised. Inshallah, you will receive the next 30 minutes of this video separately soon, in just 2 or 3 days. Till then, I want to be sure that you have watched this video and you have understood all the content which I have delivered in this particular video. Type me the answers. I want your answers in the comment section below to those questions which I have posed to you while I was delivering this lecture. Okay, my dear students, I'll be showing you a small clip for that content which you will be receiving in the upcoming video. That means your video lecture number 7. I'll show you a small clip, let you have an idea how much informative that video is going to be. Till then, take care of yourself. Fi amanillah.
Okay, fourth is your Alzheimer's disease. Alzheimer's disease. This is also, it has got multiple other reasons also, but it has got some potent autosomal dominant reason also. Okay, Alzheimer's disease. Fifth one is your achondroplasia. How can I forget this? Achondroplasia. Okay. Sixth one is going to be, what else I will give you, is myotonic dystrophy. Myotonic dystrophy. There are many others also. I don't have board. Seventh one is tylosis. You have others also like dentinogenesis imperfecta, you have aniridia, you have anonconchia digitalis. There are many disorders, but these are very important. So Huntington's chorea, polydactyly, brachydactyly, Alzheimer's disease, achondroplasia, myotonic dystrophy, tylosis. I'll be explaining these in autosomal dominant disorders. So this is not the entire genetic disorder. Chromosomal disorders are somewhere else. Okay, in this category we have to discuss what? In autosomal dominant. Autosomal disorders. Here we don't have dominant and recessive thing. In autosomal disorders, we have to discuss what? We have to discuss Down syndrome. We have to discuss Edwards syndrome. Although Down is only mentioned in your syllabus, but they are very similar. Part 2 syndrome. Okay, so Down, Edward, Part 2 syndrome. There are 3 different disorders. And in sex chromosomal disorders, and the chromosomal disorders where you have non-disjunction basically causing the disorder, extra the cases of aneuploidy basically. Here we'll be discussing Klinefelter syndrome. We'll be discussing Turner syndrome. We'll be discussing super males. We'll be discussing super females. I've already given you this classification before. So these many disorders we have to investigate. So roundabout they are going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31. There are 31 disorders which are on the board. You can type your disorder. You can write down about any damn disorder you want to learn about from my side. If I'll be knowing, well and good, I'll be sharing my knowledge with all of you people. So whatever is the next video you want to see from my side, you can type, but that should be genuine. I will suggest that you first of all start analyzing X-linked disorders. You can type any of the disorders from this category you want me. Even if just one student will suggest me something, I'll make a video on that particular genetic disorder. So whatever are the questions I have asked you, type the answers and give me the answers of all the questions which I have asked you in between the lecture. Stay tuned. Give this video a thumbs up if you like this video. Share it, like it, and comment upon this video. And don't forget to subscribe to my channel. That's very, very important. May Allah keep you happy. Stay blessed. Take care. Fi amanillah.